really appreciate the opportunity to speak. My name is Hans Keirstead. I'm the chairman of Immunis, a new company that is an immunomodulator targeting age-related immunological decline. We all know that aging is the greatest risk for disease. Every disease happens with a greater frequency as one ages, of course. And we also know that there are several key physiological degradations that accompany aging, with immune dysfunction being at the very top of the list and actually partially responsible for every other degenerative response in the human body with time. The market on age-related disease is, of course, tremendous. We have some numbers in front of us that actually only represent a small capture of these sectors of sarcopenia, osteoarthritis, macular degeneration, etc., as the number of available treatments is so few, so we've seen very little actual market penetration in this sector. Nonetheless, the clinical studies and publication are just increasing at a phenomenal rate. In both the clinical studies, these are approved studies through regulatory bodies, as well as publications in the sector, we're seeing in both of these a doubling within the last 10 years. So that's a heck of a strong compounded annual growth rate, if you will, for both the publications underscoring the clinical studies that are really leading the field forward. This has resulted in a tremendous upsurge in longevity investments. The number of companies found is just skyrocketing, just tremendously, as you can see on the left-hand side of the screen. And the amount of longevity venture capital investments is also just absolutely skyrocketing. And you can note that over the last 2 years, there has just been a tremendous amount of growth in this sector, truly logarithmic. The longevity investments have really, in this field, has been punctuated just tremendously by a few major investments that, again, speak to the desire of large companies to get into this space, large investors to get into the space, which of course speaks to the tremendous available market that can be penetrated and made liquid. So here we see just phenomenal billion-dollar investments into Calico, Cambrian Biopharma closing a $100 million Series C. More recently, we see New Limit with $105 million, $3 billion Alto Laboratories. So very major investments. This is not a small dipping of one's feet into the pond. This is a 2 feet forward jumping in full-hearted. It's interesting to note that, as a scientist myself, there are actually very few ways that longevity is actually affected and truly proven in the literature. And I've listed the most prominent here. These are really the top 5 ways in which we know for certain that these interventions in human behavior and eating, diet, etc., are very well documented to affect longevity. Physical exercise, adequate sleep, a strong, healthy diet. That's part and parcel with the next one of reduced toxin exposure. And of course, more recently, caloric restriction has entered over the last decade, clearly showing that any of these behavioral modifications can very fundamentally affect longevity in a measurable way. And I just find it very interesting as a scientist that every single one of these proven effectors of longevity work through the immune system. I've just put up a handful of just a couple references for each of these, but it is very clear, indeed irrefutable, that physical exercise, sleep, healthy diet, reduced toxin, caloric restriction, all work through the immune system to have their effects. Yet there are extremely few approved immunomodulation drugs on the market. Of course, probably the most well-known is aspirin. This is inhibiting an inflammatory NF-kappa B production, reduces leukocyte and endothelial cell interactions, very clearly working through an immune response and having a rather targeted effect on heart and some on blood pressure, but clearly affecting immunomodulation as their mechanism for saving lives and inducing longevity in their patients. You know, these we also have this other category of immunomodulatory drugs, the IMIDs, that inhibit inflammatory TNF alpha production primarily, and stimulating other agonist-antagonist actions in that system. And we see very major participation, again, Celgene here, Bayer with aspirin. Antidepressants also decreasing inflammatory cytokines that I've listed there, with major players in the field, including AbbVie and Lilly and Pfizer. And I'm very pleased to say that our company, Immunis, is also directly working on an immunomodulator using a brand new drug class and affecting efficient immunomodulation, as I'll show you, and thereby longevity. The problem that we decided to approach several years ago was immune system decline. We've studied up very extensively on the immune system, of course, working with the world's leading professors. I've been a 15-year professor myself before committing 100% time to corporate. And the immune system, frankly, declines in function, but increases in its activity with age. When you're 27 years old, you're birthing your immune system about every 2 weeks. That precipitously declines with age, meaning that your immune system precursor, born from your bone marrow, are made logarithmically less frequently throughout your life. And that has two effects. One, clearly, your immune system has to get old and tired. It's replenished less frequently. Now, that's a real problem because the immune system divides a lot. It's an extremely active organ, one that is replenishing itself constantly as you're growing up at its peak at 27. But even when the cells are not being birthed as frequently when you're through your 30s, 40s, 50s, 60s, your immune cells are certainly dividing. Now, they're not renewing each other in a rejuvenated young form, but they are highly, highly metabolically active, very active cell type that accumulate over time a great deal of both terminal damage, decreasing the immune cell population itself, but also genetic errors and other problems of their own metabolism. The second effect of this birthing of your immune system less frequently with life is the secretome, the secretions that the young immune system precursors make as they slowly mature over many, many months into functional adult cells. Like any organ in the human body, it is the young cells that do the secreting. Secretomes, the entire population of secreted factors, liposomes, exosomes, etc., from cells, everything's secreted, including exosomes, of course. That's referred to as the secretome. It's actually the secretome that we're targeting here as a therapeutic target to replace, because it's the secretome that is a much more amenable, druggable target, in that cellular replacement for the former problem, the immune system precursor decline themselves, is a much trickier business, in that the generation of cellular therapeutics, very expensive with respect to cost of goods, very prone to a poor batch-to-batch maintenance, or put another way, very prone to high batch-to-batch variability for product release, both the in-process and batch release, and the entire quality system that wraps a cellular therapy. That immune system replenishment is a very, very high cliff to climb, a mountain indeed, as a very difficult manufacturing regime with high batch-to-batch variability, poor release criteria, that type of thing for a cellular-based therapeutic, that translates directly into a very high regulatory bar. And indeed, we've seen cellular therapies in the past clinically approved, being mandated by the United States FDA to have an animal safety study accompany every clinical batch release. Just a phenomenal regulatory oversight and difficulties, making for extraordinary and frankly non-commercially viable cost of goods. So cellular therapy has many, many problems. I've been involved with them a great deal in my past lives and previous companies. But we made it through as pioneers in that field. But it's very clear from looking back on the cellular replacement field in stem cells that a secretome is a much, much highly preferred drug, as your TGF beta is the same as mine, your IL-2, your IL-10, your any protein sequence. They, even across races, are rather identical and to be distinguished from biomimetics. Many of these mimicking proteins that have been made are made in insects, and they are fundamentally different and truly immunogenic. This secretome that I'm discussing here, that we are targeting, very, very important that it be made from pure human, natural human systems, so that you don't get that immunorejection of a proteinaceous drug. So the solution with that background was to take proprietary liquids, and that's really quite a bit of the substantive patents that we have, and dump them on both clinically and commercially viable stem cell sources. We use both embryonic or iPS cells in order to do this, according to patent amenability in particular jurisdictions, or just preference of a particular country. The cell types behave differently, but we can affect these differentiation regimes using any good starting stem cell population that is equipotent in that it makes all three germ layers of mesoderm, endoderm, ectoderm, so that we can differentiate in a very directed manner to very high purity cell populations. And this has been my background. I've been a 15-year professor at the University of California at Irvine, as well as Keio University in Japan. And during that time, I became the world's first researcher to use human stem cells for anything to do with the central nervous system, actually. And some of the leading regulatory approvals in this sector, led by myself and my team, of course, that have resulted in now this being our 17th FDA clinical trial approval in this sector. So I've applied the learnings from my past to this differentiation paradigm. My group was the first in the world to take any stem cell population and differentiate it into a highly purified derivative. We started out with oligodendrocytes, a central nervous system cell type, of course, that myelinates axons, built that up, sold that to Geron Corporation, which went through clinical trials. Then high purity, again, 99.6% purity of motor neurons, sold that to Lonza Corporation. Several other that I won't go through now, but my background has all been with my team taking human stem cells clinically and commercially viable stem cell lines, and then differentiating them to extremely high purity. The rest of the field hovers around 50 to 70%. And that contaminant load, that tremendous 50 or 30% contaminant load, prohibits both cellular transplantation and secretome work, because you have a heterogeneous population producing a secreted product. So in this case, we have very, very high purity derivatives. Again, 99.6% purity. And what we do is collect their secretions, so we can grow them by the billions and farm them, frankly, to produce secretions in a very, very controlled manner that results in extremely little, as in no statistically significant variation between batches. So we're taking the secretome here. That's the total of the secreted content of the cell population, which is young and highly secretory. And that is our product, an all natural, all human set of immunomodulators in their native, their natural relative concentrations. So this isn't anything that is non-physiological. These are all physiologically relevant concentrations not only in total, but also in relation to one another. This secretome, I don't have time to get into it now, but contains 690 factors. Very, very importantly, no bad things like inhibitors of tumor suppressors, but all elements that have been identified to both regulate and promote the development and also the maintenance of the immune system, that extremely complex organ that is a yin-yang, an agonist-antagonist, extreme redundancy. To think that we can modulate the immune system with single drugs is very difficult. They do work in some cases, of course, but a secretome provides a very unique opportunity to regulate this organ in a very physiologically relevant manner, one that is very natural and therefore low safety problems. And really, it is the secretome that is our product. So the product itself, which we have called STEM, is all natural and it's all human, all physiologically relevant concentrations, as I've said. And it includes multiple effectors: transcription factors, growth factors, angiogenic lipid mediators, nucleic acids, cytokines, chemokines, hormones, extracellular vesicles, exosomes, etc., secreted components in total. And of course, that secretome serves multiple biological functions, and I've listed several of them there. This is a paradigm shift from single active drugs, which have to be in a system like the immune. They really are a 2x4, a single active, trying to overcome the great redundancies of that system. A single active trying to overcome its competing factors that naturally occur, whether that's an agonist or an antagonist. That extremely complex immune system, to throw a single active ingredient at it is one very, very tough mountain to climb. A secretome is something that can replace it in its entirety. Now, this wasn't possible several years ago because the regulatory environments of all nations were built around single drug components. That's how large pharmaceutical companies have been built. And it was only a few years ago that the United States FDA and every other major regulatory body in first and second world nations put in place a new drug category, that being secretomes. In the United States FDA, it is run under the Cellular Biologics or CBER division, and it's a new division. It's a new drug category that permits a multi-component actives drug. It's relatively new. Worldwide, I've counted 17 phase 1s that have completed. Interestingly, none of them have had adverse events, which is phenomenal. And it's also to be expected, as these secretomes are naturally occurring elements. And I do want to underscore one thing here. A secretome is not a secretome is not a secretome. They vary according to tissue types. To think that you could take naive stem cells and just take their secretome, what are you going to get in that secretome? You're going to get a stem cell specific secretion set. So wonderful if you're trying to get stem cells to either propagate themselves and grow into more, or do a gross drop out of cell cycle into a heterogeneous differentiation pattern, but absolutely irrelevant to the immune system, absolutely irrelevant to the skin, eye, gut, hair, cartilage, etc. You need a tissue specific precursor in order to generate a secretome that is relevant to the organ. So in this case, of course, ours is immune precursor. We have completed all of the science studies that relate to safety. So of course, the United States FDA and every regulatory body across the world requires third-party, arm's length, GLP toxicology laboratories in order to do your heme tox safety studies for pharmacovigilance. So we did provide our drug and dictated the experimental design with BTS Research after a pre-IND meeting with the FDA. And we agreed upon the following safety study, where we treated animals and then collected various tissue compartments and showed no adverse effects. And those included a demonstration of no adverse changes to the blood, the urine, the behavior, the body weight, the food consumption, or even gross histopathology in multiple organs. So having satisfied that, we actually, dealing with an immune product, decided to raise the bar above what the regulatory body demanded, and convened think tanks with several of the world's leading immunologists, and said, if we have an immunomodulator, what would be our worst nightmare? If it's perhaps not detected in blood, urine behavior, body weight, food consumption, and histology, what else could be happening here that we should be concerned about? And of course, top of that list was, if you immunomodulate, you may induce cytokine storm. And so we were very, very careful, of course, to run studies that demonstrated we do not cause a cytokine storm. Likewise, other studies in different laboratories showed that we do not induce adverse immune responses. And then the third category of concern, excuse me, would be enhancement of distinct pathogen growth with an immunomodulated system. And of course, we ran those studies with multiple pathogens to show that our immunomodulatory does not cause a niche, for example, of gross pathogen amplification. So safe on all factors. Then moved on to efficacy studies, both pilot and then formal studies that satisfy the FDA requirements, and that led to the publication that I've just taken a screenshot there on the right side of the screen. We took this drug and put it out in the hands of multiple leading National Institutes of Health, National Institutes of Aging professors. These are particularly world-renowned aging professors. And I handed this drug out to several of them, 5, in fact, of 5 different manifestations of immunomodulation, different manifestations of aging. And in every one of them, we saw positive signals. So this is a drug that has a very broad-reaching efficacy on multiple organs, as would be expected from a broad-spectrum secretome immunomodulatory drug. For reasons that I'll show you in a moment, we focused most on muscle degeneration or sarcopenia with aging, or muscle degeneration as a result of immobilization. So running both of those studies, Professor Drummond, so third-party laboratory, demonstrated in blinded manners the following: that our drug prevented age and disuse associated, think of a cast or immobilization, both age and disuse associated decreases that naturally occur in muscle mass, strength of the limbs, and fiber cross-sectional areas. Also prevented age and disuse associated increases in muscle fibrosis, so halting that. Increased rodent muscle cell growth in dishes. And then perhaps most importantly of all this work, showed tremendous effect on human muscle in a dish. So increased human muscle myoblasts in their division rate and their fusion, myonuclear fusion, causing myotube areas to grow. So this actually caused muscle fiber growth with a very detailed mechanism of action of myoblast division, myonuclear fusion in order to generate the increased myotube areas. In other research besides that, which was published in one of the leading aging journals, Geroscience, that's an impact factor of 8. I'll remind you, most journals have an impact factor of 1 to 3. We also handed the drug out to other leading groups. So one professor showed that it, in a series of studies, decreased age and disuse-associated increases in general inflammation and general clinical disease. Another neuroscientist showed that it decreased neuroinflammation and the manifestations of that, which are demyelination and central nervous system degeneration. In other industry standard animal models, showed that it prevented age and disuse-associated increases in arterial stiffness. Phenomenally enticing data set. Of course, arterial stiffness increases with age. Your tubes, your arteries become less flexible as they are when you're young and more like solid tubes, resulting in blood pressure changes, hemorrhagic stroke, and heart attack. That is an extremely attractive target for us. Also increasing the effectiveness of the adaptive immune response, which is known to decline with age. So very, very good data. I'm not leaving anything out. We never gave it to a laboratory that showed no effect. Interestingly, all of these efficacy laboratories did show a a difficulty keeping themselves blinded, frankly. So we had to take a lot of measures in order to keep proper blinding on. Because of the general effects on the aging animals, the treated animals were often moving more, had better hair coat. Anyone working in the sector realizes that aging mice have what's referred to as a punk rock hairdo with bald spots and really spiky islands of hair. Their hair coats got much, much smoother. Their metabolisms were increased. Fatty liver deposits decreased, and much, much more movement within cages. So interesting broad spectrum effects in every one of these laboratories, and strong signal as well. So that led, of course, these studies with the full CMC sections and the clinical protocols to a full FDA approval earlier this year, indeed May 4th, when we had a single cycle review and then addressed some comments within that month and were awarded. I'm very proud to say this was the 17th FDA approval for my team, which has been with me through 4 companies now. So this led to a very strong and quick development path, where in the first half of 2022, we were working on our IND and completed our first round of funding, which was the Series A. And that went up very, very well, identified a clinical site, IRB approval, et cetera. And now we are running that clinical trial. So we're actively recruiting in the trial right now at UCI Medical Center. And we're preparing more manuscripts for publication. And for reasons that I'll show you, we have an extraordinarily fast clinical development timeline. When one picks a particular clinical development, you look at a matrix of different factors. We chose one that has a very short clinical trial duration, leading to very fast value accretion. This means that we can get to a phase 3 quite quickly, and indeed about a year to 18 months from now. So sometime around the end of 2023, we anticipate getting phase 3 approval with this drug, as we're in phase 1B now, which should finish its patient treatments there. And then the phase 2 is similarly quick, 3 to 6 months, and then applications for phase 3 immediately thereafter. So that is an extremely fast protocol. And the company's plan is to sell off and partner that single clinical indication that I'll get to in a moment, but then continue to run phase 2s and 3s in order to then expand the labeling for the BLA-approved drugs. Our first clinical site is UCI Health, just a tremendously strong group, backed by California Institute of Regenerative Medicine, the largest funding organization for stem cell work in the world. And I was one of the founding scientists that helped get that passed several years ago, with a $3 billion initiative to be spent only in one state of California. That was just recently renewed last year for $5.5 additional billion to be spent in the state of California. CIRM model has just been the brilliance of Bob Klein and replicated in many countries throughout the world and many other states within the United States. To a lesser degree, California has been $8.5 billion over the last several years to support stem cell research. Phenomenal boost to this entire sector. And California Institute of Regenerative Medicine put up sites that provide clinical research infrastructure, which decreases the cost to participating investigators and companies. So we were awarded that. And so we're not only partnered up with UCI Health, but also the Alpha Clinics through the CIRM network in order to facilitate this work. With excellent principal investigator, Dean Wayne, and 3 other sub-investigators. So the trial's ongoing, began in Q3 just a month ago, and very, very excited to have this through the front door of the FDA. Our first clinical indication is muscular atrophy. It is a result of 3 things: immunomodulation, disease, and aging. And it is knee osteoarthritis that combines all of these things, all 3 of those triggers, causing a very acute and profound decrease in muscle loss. Indeed, you lose about 1/3 of the thigh within 4 weeks. Just a tremendous amount of muscle loss, which means a very high clinical signal and FDA-approved, validated outcome measures for it as well. It occurs in every single person. 100% of humans have muscle loss as a result of aging, and that has severe effects on the quality of life, and we're seeing a surge in prevalence of it. As I've said, on the left, you can see that all three of these triggers are present in the osteoarthritis. So although we're treating muscular atrophy, we're using KOA patients to do so. Excuse me. That allows us to go into a phase 2 with this, and also a phase 2 with pure sarcopenia or pure immobilization. Interestingly, this is extremely amenable to space, and we are working with one space agency in order to get this stem into space, because it addresses in one fell swoop both of the two detriments for astronauts, and that's cosmic radiation and lack of mobility, causing brain inflammation and immunomodulatory degradation that is long-lasting in the astronauts, impairing their function, and also muscular atrophy. Right now, they are just trying to increase shielding, which is costly and sometimes prohibitive, and trying to have exercise regimes which are showing very poor effect in space. STEM increases both muscle and decreases inflammation. So it is a very, very good drug, and we've seen some high interest here. STEM is being produced in a CGMP FDA-compliant laboratory sufficient for commercial production, and indeed, we actually have fully scaled to commercial production. A suggestion I'd make to any burgeoning company, as the only detriment you have when you do get to phase 3, when 94% of biotech companies are purchased by larger entities. Indeed, mine have been, my prior 3 company builds. The only real challenge you have then is the manufacturing commercial scale up, which you don't usually do until phase 3, concurrent with the phase 3 trial. And there is risk in that. And that's why Big Pharma is so selective at that time, because oftentimes the phase 1 and phase 2 work are done with very small batches, non-scaled. And it is that scaling that may change the drug and perhaps lose efficacy or increase safety. So having built and sold 3 companies prior to this one, all successfully, and gone through this sale at phase 3, I decided to have the team fully commercially scale preclinically. So it's an odd thing to do, but if you have the money and you have the time and the expertise, it's a very smart thing to do, as it de-risks the clinical activities. Those phase 1 and phase 2s can actually be taken with a great deal more confidence, because the drug will not change during the subsequent phase 3 and commercialization of the drug, because the method of manufacture is identical. So we just went through a 6-gallon production of this. The other advantage that it has is that you get to know what your cost of goods are very, very quickly. This is an exceedingly low cost of good product. And what we imagine happening here is that we will have this, of course, run it through the clinical trial for various manifestations of aging and immunological decline with age. One can't run a clinical trial for aging. It is not considered a disease by any regulatory environment, but you can target manifestations of aging, which we clearly are doing. So a 2-pronged approach of clinical trials in 2 different manifestations of aging. We're going to get into another one besides muscular atrophy quite soon, while doing 5 to 10 different indications in rodent models. So by the time we are up at phase 3, we've got 5 to 10 indications in rodents and 2 indications in humans, so that the buyer sees a plethora. It really is a validated, multipotent, multi-indication platform technology that has been fully commercially scaled. That is all a great big plan to set up a larger pharmaceutical company for commercialization of indication-specific drug, but also the setting the stage for off-label use of the drug for things like healthy aging. Off-label use is a very, very common and high market use of prescribed drugs. and really increases the market penetration of those drugs, because doctors know, especially after seeing several clinical trials and many, many preclinical studies, that the drug may have broader efficacy besides that for which it received its final clinical indication. So we are going broad in order to try to get to a position where we could eventually administer this drug in a very FDA compliant manner with healthy normals in order to prophylactically stop manifestations of aging. This would be something that you would take approximately every quarter. The gross margins are enormous, and we would see a subscription-based home delivery of self-administered needles, penetration under the skin like diabetics do, or a pharmacy-based distribution. The market is just absurdly huge in all three sectors of muscle wasting, sarcopenia, and autoimmune disorders, all of those things working up to an anti-aging market that's around 200 billion annually. And interestingly, there are no current therapies that are approved for muscle atrophy or sarcopenia or aging. So we are addressing a very, very open market with a first-to-market advantage, going straight through the front door of the FDA for full accreditation. So it's our goal to change the course of immune-related diseases and aging, and do so with a product that everyone can afford. So thank you very much. Appreciate your time and attention.